everyone. Thank you for taking the time to listen to my talk today. My name is Bailey Zwarycz, and I am currently the Associate Director of Scientific Business Development at Altis Biosystems. I've been with Altis for almost 5 years now, and I'm in kind of a unique position to have had a lot of intestinal epithelial expertise, and I brought that to Altis on the scientific side. And moving more towards business development now, I've really been able to bridge the gap between what we've done and what we can do. And so today, I'm really excited to talk with you about using an intestinal epithelial in vitro model to predict preclinical GI safety. So my bread and butter, like I said, is the intestine. I talk a lot about it, and so I'm excited to talk with you about it today. We know that the GI tract is one of the major organs in your body, and the schematic on the left shows kind of from top to bottom. So it starts in your mouth, hits a lot of different organs, and then it comes out the end where we have a lot of nutrient absorption and water absorption. And so the end piece is the one we'll talk about most specifically today, and it's the colon, and it's really designed for water and electrolyte absorption. The image on the right shows you the regions of the large intestine. So you have, it kind of goes up, over, and down in the ascending, transverse, and descending regions. And if you look in the schematic right in the middle, you see that there's lots of bumps in the colon, and these are called haustra, and they're really for increasing surface area to churn all the luminal contents to get all the water and the nutrients out before it's expelled from the body. And in the bottom right of this image, it shows you a cross-section of the wall of the gut. And you can see it has a lot of layers, but the one that I'm always most excited about is the innermost layer, which is the mucosa. And all those little cells on the very top of that are the epithelium. And they're what I've studied for a majority of my research, and they're called the epithelium. And you see that they make this crypt structure that kind of weaves in and out of the mucosa to really increase the surface area so we get a lot of the water and the electrolytes out. If we zoom in on this epithelial layer, we see it kind of popped out here in the image on the right, but this focuses on this epithelial layer. And this is important because it's the first line of defense to everything that you find in the lumen of your gut. And this includes food, microbiota, pharmaceuticals, and toxins. And so the image shows you a schematic of what those epithelial cells look like. They're a single cell layer, and you can see at the bottom we have what's called LGR5 positive stem cells. And these are the true stem cells in the gut. And then the epithelium in that crypt structure kind of acts like an escalator or an elevator. The cells are born at the bottom, and then they migrate up. So we have these proliferating transit amplifying cells, and then the differentiated cells towards the top. But to help with the regeneration of this epithelial layer, we're really focused on proliferative cells. These include those stem cells at the very bottom that are going to divide about every 24 hours or so, which give rise to either more of those stem cells or these transit amplifying proliferative cells. Now, these guys are going to divide 3 or 4 times as they move up and out of the crypt to those fully differentiated cells that are responsible for mucus production or absorption. Now, these guys are really important because they are essential for all the homeostatic regeneration and as well as any response to damage. And damage of the gut can happen in a lot of ways. And the list here on the bottom left is not mutually exclusive. Things like disease can cause microbial dysbiosis and inflammation, and all of that can lead to toxicity, just like inflammation can cause disease. And so there's a lot of different ways the gut can be damaged. But at the end of the day, we're looking at what is causing these cells to die and be aberrant, and what can we do about the stem cells to make sure that our gut is functioning properly. So this toxicity of the GI tract is a really common side effect of a lot of different drugs. So the image right does show a lot of adverse events that happen when we're taking pharmaceutical drugs. And these GI toxicities, or GITs is what I'll call them, is the most common. So that's at the very top of that list. Now, a lot of people have been trying to figure out how do we best predict these GI toxicities prior to going into clinical trials. And a group headed by Matt Peters at AstraZeneca outlined a bunch of drugs. There are 32 marketed drugs for any in vitro assay. And these are really targeted towards drug safety. These drugs were chosen because they have a lot of clinical in vivo data that we can compare to see how predictive are our in vitro models. And this test set has been used by this group as well as others to validate our in vitro models. And I also wanted to point out that this paper, as well as a paper written by this next group, was reviewed by the IQ-MPS Consortia, which is a really great group of industry leaders who are working on in vitro MPS models. They have also looked at utilizing the same test set for their in vitro models of GI safety assessment. So when we're talking about in vitro models, there's a lot of different ones that we can, talk about and are used in the field. The image on the right-hand side shows kind of the four major buckets that these fall into. In A, we have 2D transwells. These are nice because we can add cells to the top or the bottom of the transwells. In B, we have a schematic of membrane-based microfluidics. So here, this is a small chip that we can add flow and stretch, and this really complicates the environment and adds more physiologic relevance to that. In C, we have a traditional matrigel droplet culture. So these guys are embedded in this matrigel matrix, and these are quite easy to passage and used in a lot of labs. And finally, D is kind of like the Cadillac of in vitro models, where we have really these advanced 3D cultures where we can add tubes and flow and different cell types to really mimic what we're seeing in vivo. And so with all of these different models available, we started to think about why are our current in vitro models failing? Why are we not able to predict this GI toxicity before we go into the clinic? So some of these models are not recapitulating the human biology. So some of them use non-human models or immortalized cell lines, and they don't really specifically focus on those important proliferative stem and progenitor cells. Sometimes the results are not reproducible. Maybe when we're using a cell line for study number 1, we'll use passage 2, but when we run study number 10, we're using passage 22. And are changes happening in those cells, and are they important, is a question that I think we often don't think about. The cultures and some of these platforms are relatively low throughput. So with them being kind of cumbersome or long to use, this could limit maybe the number of compounds a group tests, maybe the doses or the replicates. And so we can be missing that with not having enough power in the studies that we're doing. And finally, these assays don't always come with consultation or expert experience when we're running the models. And so this could lead to, using the models incorrectly or having some other problems. So what we did here at Altis is we have developed a 2D transwell system. And so that's what we're going to be talking about today. So our current culture model that we use is called RepliGut Planar. And what we have here on the left is a cross section of one of those transwells. And so we utilize just the upper compartment to put the cells in, and you see them there in green and yellow. We have a nice differentiated monolayer. And we're utilizing this transwell, which we really like because in the light pink, we have our luminal reservoir, and in that dark pink, we have a basal reservoir. So this gives us really nice access to both the top and the bottom of the cells. And when we culture our cells on these transwells, we have about a 10-day culture period, where the first approximately 4 days, half the culture, we have lots of cell expansion. So those proliferative cells are present here. And in the second half, we have differentiation, where we actually have a nice barrier. And showing you some data that we do have these proliferative cells that we can mark with the functional marker EDU. And on the differentiated side, we have the major cells that we would see in vivo, including absorptive enterocytes, goblet cells, and enteroendocrine cells. And we really like this model because if we want to assay proliferative cells, we can use the beginning of our platform. And if we're more interested in assaying differentiated cells, we can wait towards the end and run the assays then. So because we're talking about toxicity and we're focused on proliferative cells, we're just going to take this first part of the culture, which we have done at Altis and named that specific culture system StemScreen. Now on this timeline here on the left, we see it's just the initial blue part for that cell expansion phase. And on day 0 is when we plate the cells. And once they sit down and become happy, they're about 50% confluent. On day 2, they'll become more and more confluent. So when they're about 75 to 80% is when we're going to give them compounds. And then we're going to allow them to grow for another 2 days, and we'll be able to fix them and see if they formed a barrier. And what we get out of this assay system is 3 downstream analyses. The first is barrier function that we'll measure by tier. The second is getting total cell number left after the assay, which we can do by quantifying DAPI staining. And finally, we'll get proliferative cell number by looking at EDU. So first, looking at the tier, this helps us answer the question of, are the cells able to form a confluent monolayer in response to compound treatment? So the way we measure tier or trans-epithelial electrical resistance is by using a probe. One of them is shown here on the left in our 96-well plates. And in the right, this is an example of what those cells look like after we switch them to our differentiation media. You see the tier starts low and increases over time. So really any register of this tier is showing that these cells are forming a barrier. We can also look at total cell number. So this helps us answer the question of what is the viability or the total number of cells left in response to compound treatment. So the way we do this is on the very bottom. We can use high throughput image analysis using open source software called Cell Profiler. The bottom left image, the black and white one, is what the image that we get off of the microscope. The image in the middle has green dots in all of those cells. And this is how the imaging software is able to quantify those cells. And on the right is what I like to call a confetti plot, which is going to just randomly color the cells, the cell nuclei, so you can generally see how many cells are left. And intercalation to quantify proliferative cell number. And here we can answer the question of how many cells contain EDU, or did they actively uptake that EDU, or were they born during the time they were given EDU in response to cytokine or compound treatment? The way we do EDU analysis, you can see on the top schematic, we add the EDU to the cell culture. EDU is a thymidine analog, so as these cells are dividing, it's going to intercalate into the DNA, just like thymidine would. And you can see in the middle image, there is a little tag on it. And once we fix the cells and we're going to analyze them, we run what's called a click-it reaction. And you can see on the right image that it adds a little fluorophore, so we can see if this cell did uptake this EDU during the time. And so this is how we can quantify this using the same bottom image analysis for a different color channel, all those cells that are EDU positive. So back to our study design, what we did is we have this very large compound test set. I will not be going through all of these compounds. We'll just pull a couple of them that I've highlighted here with the arrows. But you can see we have a long list of toxic compounds by Peter's group with varying incidences of clinical diarrhea from 40 to 96%. And then we also have an even larger set of non-toxic compounds. So these are compounds that have proven to not have GI toxicity that we're looking to make sure in our in vitro models are not causing any off-target effects. So getting into the data analyses and the results. The first compound that we looked at is called idarubicin. And you can see on the top, we have a pretty high GI tox incidence. Now, idarubicin is going to inhibit topoisomerase 2, and it can bind to DNA. And in the clinic, we're using it to treat breast cancer, lymphoma, and leukemia, and it can help reduce cardiac toxicity. So what we first did here is we looked at the EDU. So these are all the cells that intercalated that EDU. The left part of this first image is the vehicle. So this is our 100%. This is what we would expect to see. And you can see as we move to the right from that vehicle, we have less and less of those EDU positive cells. And this is suggesting that this compound, idarubicin, is causing less and less of these cells to proliferate. We can quantify this. So this image on the bottom, this figure here, shows two different donors, our donor 5 and our donor 1, with the same compound. And you see that as we increase the concentration, which is on our x-axis, we're having less and less of these EDU positive cells. We can also look at total cells. So here on the bottom, this image is the quantification of the total number of cells. So you can see that it actually takes a higher dose of this idarubicin to start causing decreases in the total cell number. So these bottom two images on the bottom right are showing less of those cells in general present. And we can quantify this as well. So what we see here on this top right graph is we're plotting both the DAPI, or the total number of cells, which is the red line, and then the proliferative cells, which we just talked about, which is this green line. Now we can subtract one from the other to get the total number of non-proliferative cells. And what I think is interesting here is you see that at this specific dose range and highlighted by the green box, we do see a decrease in the number of proliferative cells at a lower concentration, excuse me, than affected the total cells in general. And so this is saying that we're having a cell-dependent response to the idarubicin. We can also look at what concentrations of this compound caused barrier function defects. So remember here, what we're doing is saying, are these cells making a barrier at the end of the assay? And you can see where we've highlighted the Cmax on the dotted line in this bottom graph, that the normalized tier does start to drop, and at 10 to the negative sixth and higher, we actually do not have barrier function. And this is very indicative of when the total cell number is decreasing in the top graph. So from this data, we can conclude that idarubicin causes a dose and cell type dependent toxic response in these cells. The next compound we'll look at is called colchicine, and this causes tubulin disruption that leads to the downregulation of inflammatory pathways. So we use this compound to treat gout, pericarditis, and it can help reduce the risk of cardiovascular events. Now here we're looking again at total cell number. So the top right graph in gray is our total cell number. Again, in green are our proliferative cells, and in purple are the non-proliferative cells. So what's interesting about this is that at 10 to the negative sixth, we see a drop in the number of proliferative cells. However, we're not seeing any changes in the non-proliferative cell population. This is indicative when we look at the cells themselves. So we see that the blue images on the top are our DAPI staining. And at these higher concentrations, we still have some cells left. So the top right image, there are blue cells left, but there are no green EDU positive cells in this bottom right image. And when we look at tier on those proliferative cells, we're seeing that as we lose the proliferative cell capacity on the top graph, we're losing the tier ability for the cells to form a barrier in the bottom. So you see that 10 to the negative sixth on the bottom graph is where we don't have barrier function. Now, interestingly, in this set of data, we also ran colchicine on just fully differentiated cells. So these are a full barrier. There are no proliferative cells. And you see that there's no change in the ability for these cells to maintain a barrier when given colchicine. So from both of these data sets, we can conclude that colchicine selectively targets proliferative cells to reduce the cell number. And this is what the mechanism of action is, as colchicine targets cells that are in metaphase. We next looked at bortezomib, which has about a 50% GI incidence. Now, bortezomib is going to reversibly bind to the chymotrypsin subunit of the 26S proteasome. And we use it in the clinic to treat multiple myeloma and mantle cell lipoma. And what you see here is, again, on the top right, our total cell number, proliferative cell number, and non-proliferative cell number. And this is different from our previous slide, where once we hit that 10 to the negative 7 molar drug dose, we have no cells left. This is very indicative when we look at the cells themselves. So in this bottom image of the cells, no blue and no green when we're looking at the compounds treated with these high doses of bortezomib. And when we look at the cells' ability to form barriers, we do see that both in the proliferative and differentiated cells, we do not have the ability to form a barrier at about this 10 to the negative 7, 10 to the negative 6 concentration. And we do have the Cmax pointed out here, showing that our platform is a little more sensitive than the Cmax that we would see in the clinic. So here we can conclude that bortezomib non-selectively reduces the number of cells regardless of proliferative status. And this makes sense with its mechanism of action because it's going to target all metabolically active cells. So it doesn't matter if they are proliferative or not. The last two compounds that I have to discuss are afatinib and verapamil. So afatinib is an EGFR inhibitor. You can see it has the highest incidence of GI tox from our list. And when we look at what it does to our in vitro cultures, is it very reproducibly decreases the number of EDU cells. So we're decreasing proliferation in a dose-dependent manner. Now, what I like to highlight from this data set is this is done on 2 different donors, so donor 1 and donor 5. And this was actually run on more than 10 plates. So this is at least an N of 30 for each of these points that we're seeing here. And we're seeing this nice reproducible decrease with the afatinib. Now, verapamil, we've talked a lot about compounds that decrease and are toxic to our monolayers. However, verapamil is one on the list that should be non-toxic. And so here, verapamil is a calcium channel blocker, an antihypertensive drug with a very low GI tox incidence. And when we overlap this data, we see that there is no change in the proliferative cell count among both of these donors. So again, this was done on more than 10 plates per donor. And I think that this really highlights that these two compounds are nice positive and negative controls for our study. So to summarize all the compounds that we talked about, we put together a little graph. So these are the compounds on the left-hand side with their diarrhea incidences or GI tox, and the clinical Cmax that we're seeing in the clinic. Our StemScreen platform, we went to that tier data, so the barrier formation data, and developed IC15s. Now, if you're familiar with tox, you're familiar with IC50s. However, the Peters paper used IC15 to characterize their data. So if we look on the right-hand side, how we calculated these IC15s is where we saw about a change at 15% off of the vehicles. So that would be that blue dotted line right here. So you can see in the table to the left, the StemScreen tier IC50s were similar to the clinical Cmaxes. And if anything, they were more sensitive, finding toxicities at lower doses than the clinical Cmax. Now, in this Peters paper, they were looking at two other platforms, Keiko-2 cells, as well as MatTek cells. Now you can see here, these were their tier IC15s. And when we look at a compound like bortezomib, remember that didn't matter what kind of cells were there, proliferative or non-proliferative, they were toxic. And because the Keiko-2 cells and the MatTek cells have these metabolically active cells, all three platforms were able to predict that clinical Cmax and were very similar. However, when we look at a compound like colchicine that's very specific to those proliferative cells, you see the StemScreen model predicted the clinical Cmax better than the Keiko-2 and the MatTeks, which were false negatives. These two platforms do not have a majority of proliferative cells, and because of that, perhaps became false negatives in this study. And so the Peters group defined true positives as within an 80-fold of the Cmax, and so those are highlighted here in the black. And then the false negatives were greater than 80-fold off of the Cmax, and so you can see those highlighted in red. So in conclusion, I think I showed you today that the monolayer platform that we're using can help predict GI safety. I think that there's a lot of clinical relevance from this platform. We are using primary human epithelial cells, and this helps to recapitulate that in vivo physiology. We're actually using the cells isolated directly from organ donors. And the toxic concentrations that we found in our in vitro model well aligned with clinical Cmax values. And this was using a test set that was unbiased and really well received in the field. The results that I showed here are very high value. I think it's important that a platform can answer, does this kill the cells? We want kind of more than that out of a platform. And so the three deliverables that we identified do help to identify the mechanism and the function. So looking at, does this compound cause defects in barrier formation and total and proliferative cell number? And finally, this model is high throughput, so it does have a short 4-day cell culture timeline. The two other platforms that I talked about have longer culture timelines, so Keiko-2s need to grow for 21 days before you can assay them. And then MatTek's platform does take 2 to 4 weeks to differentiate and be ready to treat. And this platform is also in a 96-well plate format, so it is amenable to high throughput imaging and analyses. The future directions for this assay and kind of this R&D initiative are to investigate more cells in Altis's biobank. So Altis does have a cell biobank that allows for us to look at donor diversity. So we do have multiple donors available to us, as well as more GI regions. We're very aware that toxicity can potentially be focal to specific tissues based on the mechanism of the drugs. So are we seeing differences in the small intestine versus the colon? We actually have all 6 regions of the gut from each of our donors. So we are able to test, does something look different in the duodenum versus the descending colon? And those are questions we're excited to answer. We also want to look at other toxicity related readouts. There's a lot of interest, especially at the GMP level, looking at genetic toxicity. I think we can easily look at things like gene expression, what is changing at the gene level, and also the protein level, which we can do by immunostaining and looking at secreted proteins. And finally, we would like to investigate the long-term epithelial remodeling. So we add the compounds at this blue in the blue phase, right, where the cells are expanding. But what happens if we let them grow and we let them keep growing if they aren't toxic? Are we causing changes in differentiation? Are we causing changes in barrier function or what's secreted by those specific cells? So we're excited to investigate all of these things. So I really want to thank everyone for their time and focus during my talk. And I just really wanted to end with letting everyone know that we do have a research funding opportunity. It is up to $10,000 with our current program focus being toxicology. So a lot of what I talked about today. The submission due date is relatively soon, and we do have an online proposal submission, and I added the website here. But if you are interested, please definitely let me know. My email is at the beginning of this talk, and you can ask questions in the Ask a Question box, and those will be sent directly to my email. So I look forward to hearing from everyone and hope everyone has a great conference. And thank you again.